A very good morning to all. So today I am going to explaining question and answers in biology. I choose there are four questions in biology. So the first question I decided to explain here is describe the various types of placentations found in flowering plants. And the second question is describe the arrangement of floral members in relation to their insertion on the thalamus. And the third question is name the part of an angiospermic flower in which development of male and female gametophyte take place. And the last question with a neat label diagram explain the 7-celled 8 nucleate nature of the female gametophytes. So in the next slide, let us take up explaining the first question. Describe the various types of placentation found in flowering plants. So in this question, the main terminology we should know, that is placentation. What is placentation? So placentation means the arrangement of ovules within the ovary is known as placentation. And the main organ, the flattened cushion-like tissues, L for the arrangement of ovule inside the ovary, is called placenta. So placenta means placenta is a flattened cushion-like tissue on which ovules are attached. So this is the basic concepts related to the placentation. What is placentation and what is placenta? So both are present in ovary. So generally there are 5 types of placentation we classified. The first one is marginal here, and then axial, parietal, free central, then basal. So the marginal placentation, in this case, the many number of ovules arranged on a single placenta in the inner margin of the ovary. For example, in leguminous. So in this diagram, it clearly shows that the ovules, many number of ovules arranged on a single side. So particularly with the help of a single placenta. So in the second type, axial, in a multicarpellary syncarpous ovary. Multicarpellary, many number of carpels. So all the carpels are fused in, the condition is considered as syncarpous. So multicarpellary syncarpous ovary, placenta arise from the central axis bearing a ovules. So this is the situation of axial placentation. So here there is a compartmentization. There are 3 locules are there, and each locule carry a 2 ovules. All are attached to the center axis with the help of a placenta. So for example, this condition observed in Solanum species. So the next one is parietal. So ovules are developed in the inner wall of the ovary forming falcepta. So here the inner wall of the ovary, it bears a pair of ovules. So it appears like a falcepta. So this situation observed in cucurbit species. So this condition is considered as parietal placentation. And the next type of placentation is free central. So in a unilocular ovary, so the name itself suggests that uni means single locular ovary. Ovules are arrayed from the central axis. So many number of ovules are arrayed in the central axis. So there is a unilocular, there is no compartmentalization, there is no locule here. So this is considered as a free central. So for example, the Dianthus species. And the last type of placentation is basal. So in this condition, a unilocular ovary, like that of free central ovary, it is also a unilocular. A single ovule arises from the base of the ovary. So a single ovule arises from the base of the ovary with a single placenta. So for example, in wheat. So in the next slide, I am going to explain the second question. So in second questions, describe the arrangement of floral members in relation to their insertion on thalamus. So here there are 4 diagrams. The first one is hypogynous flower, second and third one is perigynous flower, and the fourth one is epigynous flower. So based on the position of the sepals, petals and stamens in relation to the ovary of the thalamus. So in other words, it is also explained as the position of the calyx, corolla and androecium in relation to the gynoecium of the thalamus. The flowers are classified into hypogynous, perigynous and epigynous. So in hypogynous flower, the ovary is superior, the ovary is superior, and the other floral parts are present below the ovary. So this is the condition. The ovary is superior, and below the ovary we find out the remaining floral organs. So for example, it is observed in China rose. So the second type is perigynous flower. So in perigynous flower, the ovary is off inferior. So the floral parts are present on the rim of the thalamus. So with centered ovary. So ovary is present at the exactly at the center. So on the rim of the ovary we find out remaining floral organs. So this situation is observed in rose plant. And the last type is epigynous flower. So in this condition, the ovary is inferior and all remaining floral organs are in above the level of the ovary. So this situation observed in cucumber species, cucumbers. So the next question is a very simple question, that is name the part of the angiospermic flower in which development of male and female gametophyte take place. So here the male gametophyte, it represents male reproductive organ, that is pollen grains, and it develops in pollen chamber. So pollen chamber present the inner part of the anther. So when you take the transverse section of the anther, the outermost layer is epidermis. Inner to that one there is a 2 to 3 layers of middle layer. Inner to that one there is an endodermis. So inner to that one we find out a tapetum. Again the middle layer, after that we find out the tapetum. So the tapetum, it is an resin tissues. So inner to the tapetum we find out a pollen chamber. The innermost layer is pollen chamber. So this pollen chamber, it produces a pollen grains. So the pollen grains is considered as a male gametophyte. So the female gametophyte represented as female reproductive organ, that is ovule, and it develops in nucellus. So nucellus is a part of the embryo sac. So embryo sac, it consists of a nucellus. So inside the nucellus we find out a central cell. The central cells bears antipodal cells, and then egg operators, and then secondary nucleus. So all these are present. So the nucellus is considered as a female gametophyte. So the last question is, with neat label diagram explain the 7-celled 8-nucleate nature of the female gametophyte. 7-celled, there are 7 cells are there in matured female gametophyte. 8 nucleates are present. So hence it is considered a 7-celled 8 nucleation. So the female gametophyte embryo sac develop from a single functional megaspore. So there are 4 megaspores are produced during the developmental stages. So among the 4, 3 are degenerated, 1 is functional megaspore. So this functional megaspore is undergo 3 successive mitotic division to form 8 nucleate embryo sac. So initially it produces 8 nucleate. So how this functional megaspores produces 8 nucleate stage is illustrated in the diagram. So in the first one, the A, so here we find out 2 nucleus only. So this 2 nucleus is subjected to division. So it produces a 4 nucleus, and then these 4 nucleus again subjected to division. It produces 4 and 4, totally 8 nucleus. So after producing the 8 nucleate, we can easily classify the embryo sac into micropylar end and chalazal end. So in the micropylar end there are 4 nucleus, in chalazal end there are 4 nucleus. So among the 4 nucleus of each end, both from micropylar and chalazal end, so then 1 nucleus migrated towards the center from each pole. So there are 2 nucleus present exactly at the center. So the remaining 3 in micropylar end, it act as a egg and synocytes. Together we consider as egg operators. And the remaining 3 nucleus in chalazal end, it act as an antipodals. So the antipodals are present in chalazal end, micropylar end, we find out egg operators. So this is the key feature to differentiate the micropylar and chalazal end. So micropylar end always bears an egg operators. Chalazal end always bears an antipodals. So the antipodals 3 in number, egg operators 3 in number. So in the central cell it bears 2 nucleus, hence it is considered as diploid. So this diploid condition is produced due to the fusion of 2 polar nuclei. So here totally the egg operators bearing 3 cell, antipodals 3 cell, so 3 plus 3 6, and then 2 polar nuclei, 6 plus 2 8. 8 nucleus are there in embryo cell. So there are 7 cells. How these 7 cells are identified here? Antipodals 3 cell, egg operators 3 cell. The central cell is considered as a single cell. So 3 plus 3 plus 1. So totally 7. So 7 celled 8 nucleus stage of embryo cell. So I hope you clarified.